All right, good afternoon. Let's go ahead and start for today. So again, we are getting a head start, today being Sunday, of course, but I thought I will go ahead and send you the recorded lecture for what is scheduled for tomorrow, Monday, in advance. So it's nice to have the materials available timely and preferably in advance. So we shall go ahead and do that. Makes it a little easier on me too. Um, And of course, the thing to keep in mind is your lecture exam number 1, which is coming up this coming Tuesday. That would be June the 8th. And I've sent out the study guide and all of the other logistics that apply to that one on Connect. So please let me know if you still have any questions. And I did go ahead and grade all of your lab quizzes that were turned in as well as answering any specific questions that you guys had. All right, so here we are, and this is chapter number 4 that we are looking at, and this has everything to do with the study of the cell, which in itself is a rather broad science of cytology, as you can see. So cyte, C-Y-T-E, actually stands for a cell, Latin/Greek term for the smallest unit of a living thing. Also, the basic structural and functional unit of life. And that is the definition that I tend to prefer for as far as cells are concerned, right? So cells are the basic structural and functional. So again, structure and function go together, as you can see here, anatomy and physiology, right? Structural and functional slash smallest unit of life. Uh, so at all the other organizational levels, before you get to the cell itself, you cannot consider something as being living, right? So we talked about, for example, the chemical level, right? And then, of course, the macromolecules like carbohydrates, fats, proteins, lipids, so on and so forth. And then macromolecules, DNA, RNA, right? Organelles, which are these mini organs, if you will, that make up the cell. They're not living, but once you get to a uh, fully functional cell itself, that's when life actually starts, okay? And then from that point onwards, similarly shaped and similarly functioning cells come together to form tissues. And the study of tissues is called histology, okay? Uh, Again, one of the two disciplines that are taught under gross anatomy or as subdisciplines to anatomy during the first 2 years of med school. So that's what we are looking at here, cytology, the study of cells, which of course is not possible without using an instrument called the microscope, which itself was invented in the 16th century, uh, owing to the work of a couple of gentlemen that hopefully we'll take a look at here. Uh, so microscopy is what makes the study of cytology possible, right? Using that instrument made up of a number of lenses. And the one that is most commonly used in the lab or in the clinic for that matter is the light microscope, okay? And this one produces a two-dimensional image, as you can see, and why it's called a light microscope is because it uses a light source to illuminate a slide on which you have the specimen that you are trying to examine, right? It's also called a compound microscope because it uses a number of compound lenses, convex lenses, and some concave in order to give you an image. Then you get down to the electron microscopes, and these are much more powerful, if you will, than the light microscope. They can magnify things um, much to much greater extent than a light microscope. For example, if you're studying a virus, like the coronavirus as an example, or Ebola, you would be using an electron microscope, okay? You won't be able to do that under a light microscope. Then you have what is called a TEM for transmission electron microscope. And again, this gets you a 2D image of things, all right? And then finally the SEM or the scanning electron microscope, that actually gives you 3D images. So here, this picture gives you a pretty good idea as to what those 3 microscopes are all about. If you look at the light microscope on the very left-hand side, you can see this is a two-dimensional image of a tissue. And I can straight up tell you just by looking at this that we are looking at a ciliated columnar epithelium, right? You can look at these tiny hair-like structures called the cilia, You see these columnar cells, column-shaped cells underlying there. Then the basement membrane, here is the connective tissue underneath. So ciliated columnar epithelium. So where is it taken from? Most likely from your respiratory tract. As we shall see during chapter 5, that is one of the places in the human body where you have this type of an epithelium, okay? And this epithelium has also been colored, so it appears pink. So they likely are using an H&E stain, H&E, which stands for hematoxylin and eosin staining, okay? Quite widely used in anatomy and physiology, also in microbiology, okay? So that's what you're seeing here. Now let's take a look at the similar tissue, uh, only this time you're looking at it under a scanning electron microscope. So this gives you a two-dimensional picture, right? So now you can see the cilia in much greater detail. It almost looks like grass on a prairie or something, right? Like waving about in the wind. So it's much more detailed, as you can see. Actually, this is a three-dimensional image, right? Here you're looking at a transmission electron microscope image, which is a two-dimensional image. And again, you see the cilia, which kind of looks like this tall grass growing on the epithelium. So cells come in all kinds of sizes and shapes. As we can see here, the smallest cell, for example, is your erythrocyte or your red blood cells, which are between 7 to 8 micrometers, as you can see here. The largest cell, which is almost barely visible to the naked eye without the aid of a microscope, if you have 20/20 vision, is the human egg cell from a female, right? The human oocyte, which has a diameter of about 120 micrometers, as you can see here. Cells come in many different shapes and sizes, as we shall continue to see here. So first up, we are looking at the hierarchy of life one more time. So let's take a look. The atoms, the very smallest structures here, right? Only visualized using an electron microscope. 0.1 nanometers here. They come together to form molecules, again, pretty small, like water, carbon dioxide, amino acids. Then the larger macromolecules like proteins in between 1 to 10 nanometers, as you can see here. Then come your organelles, these little microorgans, if you will, which are found inside of cells, like the ribosomes, which make protein for the cell, by the way, in between, I would say about like 20 to 30 nanometers. And then the viruses, right? You see the very smallest of viruses, anywhere between 80 to 100 nanometers. The viruses, remember, are not cells. So viruses are quite cool entities. I'm not calling them cells because they're not. They're biological entities. Very simple. They only have a genetic material like either DNA or RNA. No virus has both DNA and RNA within the same protein coat. Okay, so you have DNA viruses on the one hand, and then you have RNA viruses on the other hand. And they're surrounded by a protein coat, and that's it. That's all that a virus is. A bunch of nuclear material surrounded by a protein or sometimes a lipid envelope. That's it. Very small, very efficient, no baggage attached, but these little biological entities have the power to bring the entire world and more down to their knees, as we found out and are continuing to find out in the current pandemic situation, thanks to COVID-19, the mutated novel coronavirus that we have. All right, so uh, let's take quick notes here as well. Viruses as, as non-cellular biological entities capable of altering the course of all life. Okay, so we'll get more into that once we get to the microbiology portion here. Okay, then come the mitochondrion. Okay, again, it's an organelle about 1 micrometer. Mitochondria is where you burn glucose to release energy in the process of cellular respiration. Then most bacteria, in between 1 to 10 micrometers, as you can see here. Then the smallest cell, the red blood cells here, right? And then most plant and animal cells have an average of about 30 micrometers. Here's the largest cell in the human body, the female egg, the oocyte, barely visible to the naked eye, the ostrich egg, muscle and nerve fiber cells. Then the and then an adult human for comparison, as you can see here. So here in this picture, you can see that cells come in many different shapes and sizes. As you can see, the red blood cells, the neurons in the nervous tissue. What else? The cuboidal cells, columnar cells, right? The cartilage cells, which are kind of like a boxy shape, as you can see, and the cylindrical muscular cells or muscle cells as well. So common features to all eukaryotic cells. Remember, organisms are either eukaryotic or prokaryotic, the difference being uh, eukaryotic cells do have a well-defined nucleus and membrane-bound organelles. Prokaryote, prokaryotes or prokaryotic organisms do not have a nucleus, a well-defined nucleus, okay? So that's the difference between prokaryotes and eukaryotes, okay? So in eukaryotic cells, like human cells, you have these three. You have a plasma membrane or a cell membrane, which surrounds the outer margins of the cell, then a nucleus in the middle, which carries the genetic material, the DNA and the RNA. The plasma membrane, of course, is the fence. It's the protective boundary to the cell that maintains the integrity of the cell and also acts as a selective barrier. The cell membrane kind of decides who comes in and who goes out, okay? And then the cytoplasm, which is this jelly-like fluid filling up the inside of the cell. The cytosol is the watery part of the cytoplasm, as you can see here, has a number of ions, salts dissolved in it. The organelles are these little mini organs, if you will, that again come in two categories, the membrane-bound ones and the non-membrane-bound ones. So what are the membrane-bound organelles? Here are some examples. Endoplasmic reticulum, or the ER, Golgi apparatus, lysosomes, peroxisomes, and mitochondria. They're surrounded by a membrane. Then the non-membrane bound organelles, examples include ribosomes, cytoskeleton, centrosome, and proteasomes that do not have a membrane surrounding them. And then you also have these so-called inclusions inside of cells sometimes, which includes pigments like chlorophyll, the green pigment in plant cells, or hemoglobin in humans, red blood cells, glycogen, which is the storage form of glucose in humans, and triglycerides, again, energy storage molecules, okay? So here's a perfect example of an animal slash human cell, right? So you can see a cell membrane on the outside. There is no cell wall here, which is found in plant cells again. You see this tail-like uh, appendage called the flagellum, then these tiny hair-like cilia, right? Then you see the cytoplasm, which is found inside of within the cell membrane. Here's the nucleus. You can see it's also surrounded by a nuclear envelope on the outside. Here is something called a nucleolus, which is found within the nucleus. And again, the nucleus contains the genetic material, your DNA and RNA, as you can see. The nuclear envelope also has these tiny pores called the nuclear pores, which allow for your messenger RNA to move in and outside of the nucleus. And that is something that we will focus a little bit more on later during the lecture. Okay, that's an important mechanism that we're looking at here. And then you surrounding the nucleus, you see the rough endoplasmic reticulum. Remember, the endoplasmic reticulum is two types, the rough type and then the smooth uh, endoplasmic reticulum. The rough endoplasmic reticulum is called that because it has these tiny stud-like ribosomes attached to the outside, so it makes the surface rough. The smooth endoplasmic reticulum does not have those tiny stud-like ribosomes, therefore smooth. Then you have a mitochondria. They look like these kidney bean-shaped organelles, very easy to identify, right? Then you are looking at what are called lysosomes. These are the so-called cell's suicide kit. Cool little organelles, we'll talk about them too. Then these pink ones are the peroxisomes. Again, these are the cell's cleanup crew, as we shall discover later on, okay? Inclusions like pigments, glycogen, and all of that. Some vesicles, which are like globules containing sometimes food, sometimes waste, okay? Proteasomes here. Proteasomes, again, are some uh, organelles that have to do with the breakdown of protein within the cells, and cytoskeleton, which literally means cell skeleton, which includes things like filaments, microfilaments, microtubules, cilia, flagella. They're all made up of cytoskeleton, and they give shape to the eukaryotic cell. So here are some of the functions or physiology of cells, right? They maintain the shape and the integrity of any given cell. They get nutrients, form chemical building blocks. They get rid of waste as well. So what is the advantage of using a transmission electron microscope instead of a light microscope to study intracellular structures? You can see more detail and more resolution, okay? with a TEM. Which cell is larger, an erythrocyte or a human oocyte? A human egg, of course. Their respective sizes, 120 micrometers versus only 70 micrometers. What are the three main structural features of a eukaryotic cell? The cell membrane, the nucleus, and the cytoplasm. What cellular structure is responsible for forming the boundary of a cell? The cell membrane. Okay, so let's focus on the plasma membrane. And we are familiar with this structure, hopefully, by now. We've looked at this picture many times before. Anytime you see these, the round purple hydrophilic phosphoheads and the yellow hydrophobic fatty tails inside, you know you're looking at the cell membrane. It's an amphipathic molecule, which means the heads made up of phosphates like water. They face water on both intracellular and extracellular sides, but the fatty tails don't like water. Fats and oils and water don't mix. So there you see it. They're in the middle, right? In addition, you have these blue structures called the integral proteins, which are embedded within your plasma membrane. One thing to remember is your plasma membrane is not solid. It might look like a solid wall or something. It's not, it's liquid. The best example I can give you regarding the cell membrane is, think of those buoys, you know, those big bobbing, like spherical ball-like structures that you see on the bay if you're at the shore by an ocean or a sea or something, right? So those buoys, which are used as markers for aeronautical, for the travelers, the ship crew and people like that, right? This is exactly what these phosphate heads are like. It's a liquid structure. In other words, if you had a microscopic finger or something and you dip your finger here, all of this will disperse away because it's fluid, and then it comes together to form the cell membrane again. So this semi-liquid structure of the cell membrane is also called the fluid mosaic model, a fluid mosaic model of cell membrane, okay? And it makes sense why it's called that, because it's all fluid, as I mentioned, and mosaic is like a nice pattern, like a tiles fixed together kind of pattern, which you see here with the cell, all right? So then what are these integral proteins doing here, the blue ones? They are channels or passages for ions and molecules to move in and out of the cells, all right? You also have these so-called peripheral proteins, which are basically attached only to one side of the cell membrane. They do not go the entire length like the integral proteins. So those ones are called peripheral proteins. You also have these glycolipids, right? Which means a mixture of sugar and fat, glycolipids, and they act as markers. They identify what type of a cell that is, all right? Like ID badges on there. What else do we have here? So then here's the cytosol on the inside, right? There's a fair amount of cholesterol. These yellow little globules, these are cholesterol. So see, cholesterol is not all bad. In fact, you need it to form the structure of your cell membrane. It's only when your liver, as I mentioned before, synthesizes all the cholesterol that you will ever need. On top of that, you can ingest small quantities of cholesterol and kind of store it in your liver. The problem is most people do not eat a small quantity, right? So the animal fat is what makes up a bulk of the average American's diet, and all of that cholesterol basically causes problems with the cell membrane. It turns it solid, right? And you can see what problems ensue when your cell membrane becomes solid. Stuff cannot move in and out of it. That same gunks up your arteries, so it causes problems with blood flow to your heart and your brain. Therefore, heart disease, coronary artery disease, and also strokes in the brain. So you see all of those things here as well, right? All right, so we talked about integral and peripheral proteins, and here are the functions of what these proteins uh, have inside of the cells, all right, or rather on the cell membrane. Okay, so what are those functions? Let's take a look. So these are transport proteins. They can act as channels for ions to move in and out, right? Channels. They can act as carrier proteins where they actually carry the molecule inwards. And this looks like glucose because it's a hexagon, remember? 6-carbon sugar, hexose, that's what glucose is. So glucose channels. And also, as it says, a pump, like the sodium potassium pump, which pumps sodium and potassium ions in and out of the cells, all right? Then some of the proteins can also act as cell surface receptors for binding to hormones. Um, as you can see here, right? A hormone can bind, or a ligand can bind to a protein here. Some of these proteins, cell membrane proteins, also act as ID markers, right? So like ID badges. to tell your own immune system, your own immune cells, not to attack the cell because it belongs to you. And then, of course, some of the cell proteins act as enzymes. And what are enzymes? Biocatalysts. They speed up or sometimes slow down chemical reactions. Okay. So how do lipids maintain the physical barrier of the plasma membrane? Lipids are not permissible to water. So therefore, water dissolved substances cannot pass across this lipid membrane. What type of plasma membrane protein provides the means for moving materials? Integral proteins. What are the three subtypes? Channels, receptors, channels, carriers, and pumps, as we saw in the picture before. Okay, so now we're looking at membrane transport, which means how does stuff move in and out of your cell membrane? Well, there's two processes, as you see here: passive transport and active transport. All right, so here we will look at the difference uh, between those two processes, right? So what is a passive process? Passive means no energy needed and stuff is moving from high concentration to low concentration or downhill, which is very natural and normal, right? So for example, it's much easier for you to bicycle your way down a hill than to bicycle your way up, or walk downhill versus walk uphill, right? Same concept here. Passive process is moving stuff from high concentration to low concentration, which happens naturally. You don't need any energy to be given to this movement. Two types of passive processes: diffusion and osmosis, that we will look at here. And then you have the active processes. This is going uphill when you move substances from low concentration to high concentration, or for example, trying to climb a hill or bicycle your way up a hill. Now you're working against gravity. So it's going to take more effort and more energy. So active processes require energy. And how do you get this energy in your body for these active processes, moving stuff in and out of your cells? Of course, from ATP. ATP is your power source, your energy source for these active processes, as you can see. Okay. So let's start with diffusion. What is diffusion? Simple. Uh, Stuff moving from high concentration to low concentration. It's a passive process. Okay. Many examples. If you drop uh, some liquid, as you can see here, it's a color or a dye in a jar of water, right? What happens? Initially, it's all here, but then it starts to spread out. That's diffusion, spreading out from high concentration to low concentration. And after about 5 minutes or so, the entire jar is uniformly red colored because this dye diffused evenly everywhere. So that's what diffusion is, okay? Diffusion depends on, or the rate of diffusion depends on a couple of factors, like the steepness of the concentration gradient, which means uh, the uh, more the size of the container and the more the amount of the color that you put in, the faster it will diffuse, right? The other thing is temperature. In other words, if you heat up this jar of water, this diffusion will take place much faster, because when you heat up a liquid, the molecules start to jump all over. And so this diffusion or the spreading of the color would be much faster. So those two couple of factors control how fast diffusion takes place. So simple diffusion is happening all the time in your cells. Right now, as we speak, oxygen is getting inside of your cells by means of simple diffusion, and carbon dioxide is moving out of your cells again. Simple diffusion, all right? No energy needed there. Also some fatty acids, ethanol, and urea. These are some examples of substances. Ethanol is alcohol. It enters your cells without any problems, thus making you drunk. And then urea is a waste product, a breakdown product of protein that exits your cells by means of simple diffusion, okay? So here are some examples. You can see oxygen. There's a lot of it outside of the cell, not so much inside. So the natural tendency for oxygen molecules is to move inwards from high concentration to low concentration. For carbon dioxide, it's the exact opposite. There's more carbon dioxide inside of the cells, not so much on the outside. So it moves from inside to outside. Natural processes, simple diffusion. Then you also have something called facilitated diffusion, okay? And facilitated diffusion actually um, needs a carrier protein. And one perfect example of carrier-mediated diffusion here, or facilitated diffusion, is how you move glucose or sugar inside of your cells. So here are your glucose molecules, right? Again, hexagons, 6 carbons, C6H12O6, 6 carbon sugar. So to move this sugar into your cells, you need this carrier protein, okay, which takes up the one at a time, these sugar molecules or glucose, and passes them inside of the cell. So this is carrier mediated diffusion. It's still diffusion, but it needs a carrier, okay? All right, so next up, we are looking at something called osmosis. So what is osmosis? Osmosis simply means uh, movement of water molecules from, again, high concentration to low concentration, only this time across a semi-permeable or selectively permeable membrane. Okay, that's what osmosis is. So here we are looking at osmosis in action. All right. Of course, your cell membrane, there should be no problem identifying that structure now. Again, the purple heads and the yellow tails here. And you see a number of ions and molecules, including water molecules, right, floating all around. What is quite apparent here is, though, that water is more in concentration on the outside than inside of the cell, right? So keeping in with the laws of diffusion or osmosis, what direction would the water molecules move in? From high concentration to low concentration. So from outside to the inside. And that's exactly what's happening. So this is osmosis, the movement of water molecules from high to low concentration across a semi-permeable or selectively permeable membrane. And why is it this called a selectively or semi-permeable membrane? Because it's very selective about what moves across it and what doesn't, right? And as you can see, there are also these protein channels called aquaporins, whose job it is to allow water molecules to move inwards. So there's osmosis. Osmosis, like diffusion, is a passive process. No uh, extra energy needed by burning any extra ATPs or anything. All right, so here we are looking at three terms, which we can see at the bottom: isotonic, hypotonic, and hypertonic. So it's quite important that we understand what these actually mean, right? So first up, the prefix iso means same, right? Tonic means the tonicity, and tonicity is determined by the amount of water and the salts inside of a solution, okay? So for example, if there's a lot of salt dissolved in a body of water, it's called hypertonic. Hyper means more, right? Exaggerated, hyper. So exaggerated or more what? Salt, okay? So hypertonic solution, a lot of salt in a given amount of water. If you have about equal amounts of water and salt, equal concentration, it's called isotonic. Iso means same. And then if you have a lot of fresh water and a lower amount of salts dissolved in it, that's called hypotonic. Hypo means less. So less salt, more water, hypotonic. Okay. In other words, uh, salt water or seawater is hypertonic, lots of salt in a body of water, and fresh water, rivers, lakes, ponds, streams, is fresh water is hypotonic water, fresh water, because again, more water and there's less salt dissolved in it. So hopefully that makes it easy for you to remember which is which. Also, remember, one of the most popular IV infusion fluids used in hospitals and clinical settings is something called normal saline, which is what? 0.9% NaCl. This is the isotonic concentration of solution in the human blood, okay? uh, Closely paralleling what you see in ocean water. Similar type of concentration here, okay? All right, so the question is, what happens to your cells if you place them in certain solutions, okay? So at this point, let's take a look at a forensic situation. Actually, one of my professors used this example in our third year of med school, because that is when we study forensics. And forensic medicine is the legal aspects of medicine, also called medical jurisprudence, okay? How does law relate to medicine and vice versa? All right, so here is the situation. You're working as an apprentice with your a coroner or your medical examiner. That's the guy who opens up dead bodies, trying to determine the cause of death in unexplained and unforeseen and unusual circumstances, all right? So let's say one night you are on call and your senior resident in forensic medicine is not there, and you happen to receive two bodies, okay? And the same gender, same sex, same age, very similar appearances or phenotype, right? However, there is a difference as far as the circumstances of death are concerned. So one of the bodies was that of a person who was taking a stroll by the uh, stream side or pond side in a local park and lost his or her footing and fell into that rather deep body of water, did not know how to swim, and in spite of everyone's best efforts, ended up drowning there and was subsequently fished out of the lake, okay? The other body is from a person who was on a luxury liner on a cruise in the ocean, and he was admiring the stars at night or something on deck, lost his footing, fell into the ocean again, could not be saved in time, and drowned. Okay, so you have these two bodies lying in front of you on slabs, and on that night you are woefully low on your medical equipment. So the only thing that you have at your disposal is a light microscope and some scalpels, which are knives for cutting into human flesh or tissue, of course. And your job is to figure out which is a case of freshwater drowning, which one is a case of seawater drowning, okay? So what are you gonna do in these circumstances? So you have a couple of options available. Number one, you can just surreptitiously taste each of the bodies, just take a taste, lick, and see what they taste like, salty or freshwater. I'm just kidding. Don't do that, all right? I don't think many people would be inclined to do that anyway. So the next thing that you do is, let me take a quick note here. Let's call it salt water versus freshwater drowning forensic case. All right, so the next best thing you can do is you take your scalpel and you make some cuts into some of the visible blood vessels of these two victims of drowning, withdraw some blood samples, put them on a microscope slide, put them under the microscope, and take a look at their cells, okay? And that will tell you a whole lot about what went on with these people. So first up, um, what you're looking at in this picture is the cells when placed in isotonic solution, which means equal amounts of salt and water, both inside of the cell and outside the cell. So what's going to happen? Equal amounts of water are moving in and out at any given time. Your cells are in a nicely biconcave normal shape, as should be the case right now, hopefully in all of us, like this, right? However, in one of the people that you're looking at, and let's call him person A, if you look at person A's blood cells, they look all shriveled up, right? They're all like kind of um, all shrunken and look misformed, misshapen, and many of them bursting open, okay? What you're looking at is something called crenation or shrinkage, basically. So what does that tell you about what happened with this person's blood? This person was inundated in a lot of salty water all around the cells. And remember, which direction does water move? From fresh water to salt water, from high concentration of water to low concentration of water, right? So since there was salt water all around, it sucked out the fresh water from the cells. The cells shriveled up and look like this misshapen shape that you're looking at, something called crenation. So this cell was placed in a hypertonic solution. Remember, hypertonic equals salt solution, right? This is a case of seawater or ocean drowning. You know that. All right. When you look at person B's blood cells, what you see is the opposite picture. The blood cells are rather ballooned up, big, large, and bursting open, right? They're popping open, and blood is, or hemoglobin is spilling out of them. What are you seeing here? You're now, and why does this happen? This is something called hemolysis. Heme for blood, lysis for breaking down. So hemolysis literally means your blood cells popping open, right? And why did that happen? Because this person's blood was obviously inundated in a lot of fresh water around here, right? So where does water move again? From fresh water to salt water, from hypotonic solution into hypertonic solution. So since there was so much fresh water from the pond or the lake or the stream surrounded this person's cells, that water moved into the more salty water inside of the cells, causing the cells to pop open, balloon up, and then just burst. Okay, so this type of picture is when you place your body cells, your red blood cells in this case, in a hypotonic solution. So lake water drowning or freshwater drowning and saltwater drowning. Hopefully now you will remember which is which and how this thing works. Okay, next we are looking at active transport. Okay, and what does active transport mean? Now you're moving things not naturally as they would from high to low concentration, but in an opposite direction, from low to high concentration. In other words, you're trying to go uphill, which is a more energetic task, more energy intensive task. So you need to provide energy in the form of burning ATP to power this whole process. Okay, so what are some examples of active processes in your body cells as we speak right now? Uh, Ion pumps that we are looking at here in this picture. So this is a sodium potassium, well, this is a calcium pump as we can see. So what is this pump doing? Let's take a closer look. It's taking calcium from inside of the cells where they're less in number, and it's pushing them against the concentration gradient outside of the cell where there's already so much more calcium. So it's like trying to break down the doors against 50 people, only one person or two people trying to push the door against 50 people. That's what active transport is. Only a few calcium ions here, and you try to push them against so many on the outside, active transport. And see, you need to burn ATP to get energy to power this active process that you're seeing here. So that's one example. Another example of an active process is something called the sodium potassium pump that you see here. Sodium potassium pumps, by the way, are found throughout your nervous system, on your nerves, on your cells also, nerve cells. And what they do is they maintain the electrical charge in your nerves so they can fire electrical signals in the form of currents, electrical currents, so that you can move and think and do all of your nervous system things, right? So what do the sodium potassium pumps do anyway? What they do is they pump out 3 ions of sodium, as in this picture, and they replace those 3 ions, sodium ions lost, with 2 potassium ions brought in. So there you go. That's your sodium potassium pump. All right. 3 sodiums out, 2 potassiums in. Simple. So what does that tell you about the net charge on the inside of the cell? Is it positive or is it negative? Since you're pushing out 3 positively charged sodium ions and replacing them with only 2 uh, positively charged potassium ions or cations, the net charge inside of your cell is negative, as you can see in this picture. All right, so active processes, that was primary active transport, which means you got your energy from ATP, your energy for moving stuff uphill or against the concentration gradient by using ATP. So here you see ATP with the ion pumps, right? And here again, you see ATP with the sodium potassium pumps. Okay. Now, you also have something called secondary active transport, where, why is it called secondary versus primary active transport? Because in secondary transport, you do not burn or use up ATPs to power this active process. Rather, you use the energy of some other molecule moving at the same time, and you hitchhike or piggyback on that energy to move uphill, okay? And there's 2 types of those types of secondary active transport: symport and antiport. Sym means together, same direction, symporters. Antiport means in opposite directions. So let's take a look at some examples here, all right, of secondary active transport. So what we are seeing here in these terms of these purple spheres that you see, those are sodium ions, okay? And the green ones are glucose or sugar. We talked about that before. And this is a symporter. This protein channel is called a symporter. What does that mean, symport? Moving in the same direction. Okay, so how does this whole thing work? Right, let's take a look. Look at the number of sodium ions on the outside of the cell. A whole lot of them, right? Look at inside, only 2. So naturally, which direction will the sodium ions move, keeping in with the laws of osmosis or diffusion? They're from high to low concentration, and that's exactly what they're doing. All these purple spheres are moving from outside to inside, right? So no additional energy needed here. However, look at the glucose now. Where is there more glucose molecules as compared to the other? There's more glucose molecules on the inside than on the outside. But this lone solitary glucose molecule is kind of piggybacking or sneaking in along with the naturally moving sodium ions and moving up the concentration gradient from where it's low in concentration to where it is high in concentration, right? Active transport and secondary active transport, because this glucose molecule did not burn up any ATPs to get that energy for going uphill. Rather, it just piggybacked along with the sodium ions and moved in that direction. So this is symport. Okay, now let's take a look at this example now. Here again, you have sodium ions, more on the outside, less on the inside. At the same time, you have, these are hydrogen ions, the ones in light blue. So you see that there's less hydrogen ions on the inside, but there's more hydrogen ions on the outside, right? So let's follow the movement across this anti-porter, anti in opposite directions movement, okay? So the sodium ions move in as they should from high concentration to low concentration, no problem there. But look what the hydrogen ions did. They used the moving energy or the kinetic energy of these sodium ions moving naturally here to move in the opposite direction from less to more. Symport, moving in opposite directions. Again, these hydrogen ions simply piggybacked and used up the energy of the naturally moving sodium ions to move in the opposite direction. Secondary active transport, okay? And then yet you have some more examples of active transport like endocytosis and exocytosis. What does that term mean? Exo means outside, like exit. So exo and cytosis, cell. So moving out of the cell, exocytosis, okay? And what that entails is what you can see here in this picture. Here is a secretory vesicle, which is filled up with some, I don't know, waste or broken down viruses, bacteria, who knows. But this needs to be disposed of. This is the garbage can, the trash can of the cell. So this secretory vesicle moves all the way to the cell membrane. Here's your cell membrane. It fuses with the cell membrane and it pops open and it releases the waste or whatever needs to be gotten rid of on the outside. Exocytosis. This needs ATP. So this is an active process. All right. Similarly, you have the opposite of exocytosis, which is endocytosis. What does that mean? Endo, taking in. So endocytosis has many different forms as well, one of which is called phagocytosis, as you see here. Phagi in Latin / Greek means to eat. So phagocytosis literally means cell eating. So how do cells eat? And what types of cells eat anyway in your body? Your immune cells, your white blood cells, literally they are like these little Pac-Men, right? Let's call them the Pac-Man cells of the body. And they go around, they're the cleanup crew, and they go around cleaning up all the debris and waste that needs to be cleaned up. All right, so how do they do that in phagocytosis? This is what happens. Here's a cell. You can see the cell membrane. And the cell actually extends these two tentacle-like extensions, right? They almost look like tentacles. But these are called pseudopodia. Pseudo means false, pods means feet. So false feet, pseudopods. And so these two extensions are basically used to embrace the molecule that they want to take in, right? And then it's pulled back in. Phagocytosis. Amoeba, which are these single cell protists, they get their food the same way. That's biology 101, high school biology, right? So pseudopods being used in phagocytosis. And another example uh, of active transport. Then you have penocytosis. Peno means to drink. Phagi was to eat. This is cell drinking. So what happens in cell drinking or penocytosis? So this should be penocytosis, not phagocytosis. That's a typo. So, all right. So what happens here is there's a bunch of molecules that the cell wants to take in. Penocytosis is a type of endocytosis, taking stuff in. And so this stuff is floating about and it lands on the plasma membrane or the cell membrane. So what happens? The cell membrane kind of pinches inwards. You see how it's forming kind of like a pouch inwards? And then this pouch actually breaks off and moves in. So this is cell drinking. Rather than moving your legs or your pseudopods out, as in phagocytosis, you're simply like sinking in and breaking and pinching off and moving into the cell. And this is called penocytosis, okay? Yet another type of endocytosis is called receptor-mediated endocytosis. So what does that mean? There are certain receptors on your cell membrane, such as this green stuff. These are receptors, proteins, which act as receptors. They bind to a certain substance, and then they pinch off and move inside of the cell, all right? So one important application of this receptor-mediated endocytosis is what we see in FH. FH, which stands for familial hypercholesterolemia, which means you have the genetics because you have a family trait, the genes from your family for having high cholesterol and bad type of cholesterol. And as we shall see, the bad cholesterol is what LDLs are. LDL stands for low density lipoproteins. They have low density because they're packed with lipids or fats. So bad for your health, right? So these LDLs basically they attach to these receptors, the LDL receptors on your cells, and that's how your cells take up all of the bad cholesterol in, right? And you need this LDL to be inside of your liver cells, because if your cells don't take this LDL in, this bad cholesterol is going to go and clog up your uh, arteries, and that's the problem. So, but people with familial hypercholesterolemia lack the receptors for binding to these LDLs. Therefore, they cannot imbibe or ingest. The cells cannot take in LDLs. Therefore, where does this LDL go? It deposits all over their body, in their blood vessels. They're prone to forming these fatty plaques on their eyes called xanthomas. And I'm going to write it down here. Xanthomas, cholesterol plaques, right? Which is a sign of possibly familial hypercholesterolemia, certainly elevated levels of cholesterol, right? So xanthomas, cholesterol, uh, plaques, right? They form on like on your forearms, on your eyelids, and those kinds of places. So greatly increased risk of heart attack, strokes, atherosclerosis, which means your arteries become stiff and thick with cholesterol, all kinds of things. Okay, how does oxygen and carbon dioxide diffuse out of a cell? By simple diffusion. Compare and contrast how an ion like sodium is transported across the plasma membrane versus how a glucose is. Sodium can move in and out uh, through a channel by means of passive diffusion, no energy needed. Um, Glucose needs a carrier protein, so that's facilitated diffusion. Again, no energy, this is passive too, but glucose will need a carrier protein. Osmosis is the diffusion of water molecules from a high to low concentration across a selectively or semi-permeable membrane. What occurs to the tonicity of a cell when it is placed into an isotonic solution? Nothing, it stays the same. It's a healthy balance. A hypotonic solution, remember what is hypotonic? Fresh water. So what's going to happen to your cell? It's going to balloon up and pop, hemolysis, because water moves from fresh water to salt water, from uh, from hypotonic into hypertonic solutions, or a hypertonic solution. In a hypertonic solution, like in a seawater situation, your cells will shrink, something called crenation. What general conclusion can you make concerning the movement of water? Water moves from high to low concentration, like anything else. There's always a net movement of water by osmosis towards an isotonic, a hypotonic, or a hypertonic solution, towards fresh water to to salt water, so towards a hypertonic solution. What process, two-step process involved in the movement of sodium down its gradient into to power and the substance up its gradient. So that would be like anti-port movement, anti-porter, right? Engulfing of a bacterium by white blood cell occurs with what type of cellular transport? Phagocytosis, or cell eating. All right, so next up we are looking at our resting membrane potential. All right, so what is the resting membrane potential? The resting membrane potential is the electrical values or the voltage that is found inside of your cell on a regular basis. That's what it is. And what maintains it at that level, which is, as you can see here in the voltmeter, is negative 70 millivolts. And what maintains it at that level is these numbers of ion channels, all right? So for example, your sodium potassium ions or sodium potassium pumps, your sodium ion channels and your potassium ion channels, sodium leak channels, potassium leak channels, also sodium potassium pumps. Altogether, they determine the resting negative 70 millivolt value inside of your cells, all right? And so how does that happen? Well, what did I mention about the sodium potassium pumps? For every 3 sodiums that are pushed out that you can see happening in this picture, only 2 potassiums are brought in. So that obviously gives rise to a negative internal cellular charge, right? On top of that, you also have certain negatively charged proteins inside of the cell, like phosphates, which are negatively charged, all right? So, so this is what it usually is. At the same time, you also have these sodium and potassium leak channels. So when the sodium leak channels open up, what happens? All of the sodium from outside starts moving in, right? Because they were being kicked out by the sodium potassium pump. They're all gathered up here waiting to get back inside. As soon as the sodium leak channels open up, they start moving in, causing the insides to become more positive. And remember, when the insides of a cell become more positive, that is called depolarization. Depolarization happens by means of the sodium ions moving in through these leak channels. Okay, all right, so depolarization. But what happens when the sodium leak channels close and these potassium leak channels open up? Then the excess potassium ions, which were inside, they start moving outside. So when the positively charged potassiums start moving outside, the insides become more negative, and this is called repolarization. Okay, so remember, sodium moving in, inside becoming more positive is called depolarization, and potassium's moving out, the inside's becoming more negative, that's called repolarization. Something to remember here. Okay, so, and that's pretty much what we are looking at here as well, the role of potassium and sodium ions, okay, as well as the resting membrane potential, okay. So what is resting membrane potential? The electrical voltage across your cell membrane under resting conditions, which is about negative 70 millivolts. How is that established and maintained? By means of your sodium potassium pump, sodium leak channels, and the potassium leak channels. Okay. All right. So here we're looking at three examples of how different cells communicate or talk to each other. Okay. And remember, we talked about cancer cells. Okay. And one way that cancer cells are different from normal non-cancerous cells is that cancer cells have lost something called contact inhibition. Okay. That's what has happened with cancer cells. So the question is, what is contact inhibition? Very simple concept. When cells start multiplying or dividing and two cells come in contact with each other physically, they stop growing because it's a signal to them that there's too many of cells all around. So they stop growing, right? It's like if you're building your fence and it's touching your neighbor's fence or whatever, then you need to stop building because then you're encroaching into someone else's territory, right? So that's basically what contact inhibition is, which your cells demonstrate normally. Cancer cells have lost contact inhibition, which means they will go right over the surrounding cells and build on top of them and around them and everywhere. It's like this, uh, again, very, very like demented and predatory type of neighbor who does not care about your privacy and is constantly building his stuff on top of your stuff, right? That's what cancer cells do. However, these cells that we're talking about here do not show this behavior like your immune system. It's extremely important for immune systems to be able to distinguish normal cells from unwanted cells because guess what happens if they can't make this distinction, then your immune cells will start attacking their own body cells. What have you developed now? An autoimmune disease. Or if they are so uh, pacifist that they do not even attack unwanted cells like viruses and bacteria, what do you have now? An immune deficiency disorder like HIV infection or something, right? Sperm and egg, that is another example of two cells that actually talk to each other. And studies have shown that uh, the sperm, which is the healthiest and most robust, actually responds the best to these chemical signals from the egg and outswims and outcompetes all of the other millions of cells competing with it and makes it to the egg, and the egg kind of welcomes it inwards like a welcome guest so fertilization can take place. All right, so there's a whole lot of pheromonal uh, chemical communication taking place even at that level when a sperm is trying to fertilize an egg. And then cellular regrowth following injury. So once you have an injury like a cut or a scrape on your knee as an example, and so then uh, cell division starts happening to patch up your skin here, okay? And contact inhibition, as soon as they come in contact with each other, they stop growing at that time. Okay, so ligand receptor signaling. So how do cells talk to each other? They talk to each other using these molecules or neurotransmitters called ligands, all right? And there's three types of receptors that do this binding that are called channel-linked receptors, enzymatic receptors, and G protein coupled receptors, all right? So what are channel-linked receptors? As you can see here, receptors that are attached with a channel. So once the ligand or the neurotransmitter binds here, like a lock and key, it opens up the channel and the stuff can start moving in, okay? Um, enzymatic receptors. So once the ligand or the hormone or the neurotransmitter, whatever comes and clicks into place here, then this enzyme gets activated and it starts to phosphorylate or add phosphates again within the cell. And so that can affect the activity of the cell as well. And finally, you have the G protein coupled receptors, which is a longer process. So once the ligand binds to the cell, then GTP, which is a second messenger, binds to the G protein, causing G protein activation. The activated G protein actually moves on to activate the protein kinase enzyme, and then the protein kinase enzyme actually causes phosphorylation and the enzyme activity. So there's a rather long circuitous process that you see here. So what are some examples of how cells communicate or talk to each other? Sperm and egg, immune cells, and also your cellular regrowth following injury. How do the actions of enzymatic receptors and G protein coupled receptors differ? Enzymatic receptors are shorter, right? As soon as the ligand attaches to the receptor, the enzyme activates. Protein coupled receptors has way more steps than that, as we saw. Okay, let's talk about the individual organelles inside of a eukaryotic cell. Let's start with the ER. ER not as an emergency room, but as an endoplasmic reticulum, all right? This is one of the membrane-bound organelles. It has a membrane on the outside. It comes in two shapes and forms, rough ER and smooth ER. We talked about those two types before, right? So here you're looking at the rough endoplasmic reticulum under the microscope, and you can see why it's called rough. Look at the outer layer here. You see those little stud-like things attached to it called ribosomes. It gives it a studded, rough appearance, therefore rough endoplasmic reticulum. So what does this reticulum do? What do ribosomes do for that matter? They make protein. So protein synthesis is happening here. And once the protein is made by the ribosomes, it is transported within these endoplasmic reticulum. It's kind of like a passage, a highway of channels and alleys that transport protein, freshly synthesized protein all across the cell. So then you have the smooth endoplasmic reticulum here. It's called smooth because this one does not have the ribosome studs attached to it. So what does the smooth endoplasmic reticulum do? It is responsible for detoxification, detox of drugs and stuff inside of the cells. It also synthesizes steroids and fats or lipids, all right? And so you can look at those functions here in this picture as well, all right? Then you have the Golgi apparatus. And what is the function of the Golgi apparatus? You can think of the Golgi apparatus as the post office of the cell, all right? The post office of the cell. Let's take notes here. In other words, the Golgi apparatus modifies, packages, and sorts out proteins, like sorting out mail and sending them out to the specific addresses, right? So therefore, the Golgi apparatus would be most akin to the post office of the cell, all right? It'll help you remember. All right. So then moving on, here you can see the Golgi apparatus, all right, on an electron micrograph, and you can see how the proteins are being packaged and modified and sorted out and then sent out to where they're needed. Lysosomes. So these were called the suicide kits of the cell, right? Rather colorful term. So suicide kits of the cell. And why they're called that is because lysosomes have these hydrolytic enzymes or cannibalistic enzymes, which are able to eat the cell itself, right? And so lysosomes are basically there in order to break down and digest harmful materials that might enter your cells, like viruses and bacteria. So the hydrolytic enzymes within them will go ahead and break down bacteria and viruses and other pathogens into, well, just rubble, right? So you can get rid of it by exocytosis. But sometimes when there's damage to the cell, and what can damage the cell? Either physical insult or injury, right? Like temperature extreme, and you get like a frostbite or something, that is thermal injury to the cell, or chemical injury. If you poured some strong acid on your skin or base, that's chemical injury. Electrical injury, if you got got struck by lightning or a high voltage electrical bolt, something like that, it's going to cause electrical injury to your cells. Whatever the case, these lysosomes pop open, release their cannibalistic hydrolytic enzymes, which will end up self-digesting the cell and killing the cell, recycling it, all right? Peroxisomes. So these organelles, if you look at the name peroxisomes, it comes from peroxide, and that's what they release. They release peroxide, and we all know what peroxide is used for disinfection, right? And that's exactly what these organelles do. They disinfect the cell, all right? They also have a role in chemical digestion, lipid synthesis as well, and detoxification, okay? So at this point, let's look at a clinical picture of a disease called the Tay-Sachs disease, which is quite common in certain ethnicities, like the Ashkenazi Jews, right? A greater than average number of people of Ashkenazi Jewish inheritance tend to carry this trait, Tay-Sachs disease, because again, in some of these communities, close marriages and marrying within the clan and interbreeding is something that is seen generation after generation, and therefore some of these genes tend to get amplified in generations that intermarry, okay? So what happens in Tay-Sachs disease is, as you can see here, it's a lysosomal storage disease, which means First of all, it's genetic, as we just mentioned. These people lack an enzyme genetically, which is needed to break down your membrane lipids or fats. So since you cannot break down the lipids in the cell membranes, it accumulates within your nerve cells. Your nerve cells become dysfunctional because there's too much lipid accumulating there. Paralysis, blindness, deafness, mental retardation, and death by age 4 is what you see here. Okay. Mitochondria, next up. Mitochondria are the power generators, the power plants of the cell, right? And why are they called that? Powerhouses of the cell? Because this is where oxygen is burned with rather glucose is burned with oxygen to release energy in the form of ATP. So remember cellular respiration, glycolysis, intermediate stage, Krebs cycle, citric acid cycle, then the electron transport chain, all of that takes place here in the mitochondria. Ribosomes, again, ribosomes are protein makers of the cell, right? The bound ribosomes are attached to your smooth endoplasmic reticulum, as we mentioned before. And then there are also free ribosomes, which synthesize or make protein for the cell again. So here in this picture, you can see the bound ribosomes, which are attached to the rough endoplasmic reticulum, and also the free ribosomes swimming around in the cytoplasm. Centrosomes are these, they are part of the cytoskeleton, first of all. And what centrosomes do is they help the cell divide. When the cell divides into two daughter cells during mitosis, the chromosomes actually move equally apart to the two daughter cells by means of these organelles called centrosomes. So they're used for cell movement. Proteasomes are, again, another example of non-membrane bound organelles. What they do is they digest and break up proteins which are not needed. As we get older, our proteasomes don't function as well like the rest of the body. And when that happens, all of these toxic proteins, if you will, they start accumulating in our system. And so that's how aging happens as well, all right? Proteasomes is what you're looking at here in this picture as well. All right. So then cytoskeleton is next up, which is a non-membrane bound organelle again, as we see here. And cytoskeleton has all of these functions. It functions in cell division, like we talked about about centrosomes and so on, movement of materials, intracellular transport, and all that. And there is 3 types that make up the cytoskeleton: the microfilaments, the intermediate filaments, and the microtubules. So the microfilaments, some examples are like actin, and this is a type of protein found in muscle cell, okay, in muscles. Intermediate filaments, which are found on cell junctions where the 2 cells meet together for structural support, and microtubules, which form things like flagella and cilia, like these tail-like structures, flagellum, like the sperm, human sperm has a tail, that's a flagellum, or hair-like cilia, that's microtubules, all right? So here you can look at the microfilament, like the actin in the muscle fiber, microtubule, and then the intermediate filament here, okay? So there we go, cilia, flagella, these are all examples of cytoskeleton structures that help with cell movement, with absorption of materials and all those kinds of things. So here you're looking at a microvilli, these little finger-like projections which are found in your small intestine. And what they do is they increase the surface area available for absorption of food materials there. Membrane junctions, which means where 2 cells come together, 2 cell membranes come together, and how they are held in place. So you have tight junctions, all right? And tight junctions are called that because the junction is so tight that stuff cannot move in and out easily from here, all right? Then desmosomes, and desmosomes are halfway between tight junctions and gap junctions. So gap junctions are where there's large gaps in between the junction and stuff can move easily in and out. Desmosomes are in between. They're not as tight junctions, but they're not as loose and gappy as gap junctions either, as we see here, okay? So here you're looking at these 3 examples of membrane junctions, all right? So tight junctions, see, they're tightly bound together, not much stuff that can move across. An example would be your skin. Your skin has lots of tight junctions, and that's why your skin is waterproof. We know that. Skin is waterproof thanks to these tight junctions. They don't allow much water to escape. Then the gap junctions, such as what are found in your heart cells, in your cardiac cells, right? It's called gap junction because there's a huge gap, and stuff like ions can move easily in and out, which is something needed in the functioning of your heart. And then finally, desmosomes. And desmosomes, as you see here, are halfway between tight junctions and gap junctions. And they are also found in places like in your subcutaneous tissue, the tissue that lies just below your skin, okay? So describe the general structure of both the endoplasmic reticulum Golgi apparatus. Well, both of them have to do with protein synthesis, modifying, packaging, and sending them out, okay? Lysosomes and peroxisomes are both small membrane enclosed sacs. Which of these functions is both digestion and synthesis of molecules, okay? So both digestion and synthesis of molecules would be peroxisomes, and digestion of unwanted organelles like suicide kit, lysosomes. Which non-membrane bound organelle functions to digest unwanted proteins, lysosomes. Form the structural support of cell membrane. Synthesize proteins, ribosomes, and participate in cell division, centrosome. Which cellular surface structure functions in increasing the cell surface area, villi and microvilli, moving material past the cell membrane. Which cellular junction provides resistance to mechanical stress, desmosomes, allows the passage of ions between cells, gap junctions. And it says, let's see, hold on. Oops, where did it go? There. No, sorry. There. Prevents leakage between cells are tight junctions, right? Okay. All right, next up, we are looking at the nucleus, the brain of the cell, if you will, in the eukaryotic organism, in the eukaryotic cell, because all of the cell's machinery is directed from here, from inside of the nucleus, right? So the nucleus is the circular structure, membrane-bound organelle in the middle of the cell. It's surrounded by a nuclear envelope, which has these holes called the nuclear pores in it. And inside of the nucleus, you have DNA, which is the cell, and RNA as well, which are the cell's genetic material. You also have this another spherical body embedded within the nucleus called the nucleolus. And this contains RNA, ribosomal RNA, or rRNA, and protein, okay? So at this point, let's talk about the genetic basis of life, which is this wonderful molecule called DNA. And this double helix structure, which is still quite mysterious, to be honest, right? We have studied so much about the DNA, but there's so much more to be understood. Things like 97% of human DNA is something called junk DNA, right? Meaning it has no known function, but it does have many important functions. Some of them as yet undiscovered percent is junk DNA, right? So we are still barely scratching the surface. But back in 1952 was when two scientists, Francis Crick and James Watson, they came together. Francis Crick was British, James Watson is American. He's still alive, interestingly, by the way. He's like 96, 95, 96 years old, lives somewhere on the eastern seaboard, Pennsylvania or Maryland, one of those places, and is still active in his DNA lab, okay? And he has some colorful ideas too. And so I highly encourage you to look him up on Wikipedia, James Watson, all right? So together they cracked the double helix structure of the DNA back in 1952, for which they won the Nobel Prize in Medicine and Science in 1962, I believe, okay? However, they were so intrigued by the structure that they had discovered that Francis Crick explicitly stated that it is more likely that a hurricane or a tornado passing through a metal junkyard or a scrapyard will assemble a fully functional jumbo jet, then it is likely that the DNA molecule arose by itself spontaneously, right? That's how unlikely it is that DNA just came into being by itself. It's such a sophisticated, complex, intelligent molecule, for lack of a better term, that it just could not have arisen spontaneously. In fact, not James Watson, but Francis Crick mentioned in a book that he published in the 1960s called Intelligent Life, Francis Crick's quote on DNA and book on ET origin, let's call it that. Right. So in this book that he published called Intelligent Life in the Universe back in the 1960s, or was it in the 1970s, sometime during that time period, that he claimed that it seems like DNA was actually bioengineered by some alien race, which is much more um, advanced than the human race of the time, and then uh, consciously seeded on planet Earth for life to evolve. It's a fascinating idea. So many science fiction movies and stories and novels and whatnot have been based on that idea. Something called panspermia. In fact, directed panspermia, right? It's a type of panspermia which states that life was, or life building materials like DNA were brought to planet Earth uh, on meteors from outer space, right? Directed panspermia goes a step further and claims that some intelligent alien civilizations have done that. It sounds like a preposterous claim, but if you study DNA in its detail and follow the evolution of life and how life has gone extinct and then come back at random points in time during the previous 5 mass extinctions, you start to wonder, how is this happening? All right, so it doesn't seem that preposterous when you really get into the depths of that. So something that I've thought about myself a whole lot uh, over the past few years as well. All right, so, and you can see why we would think of the DNA as this mystical molecule, right? So first up, what is it made up of? It's a double helix structure, like a twisted ladder, as you can see here, right? The backbone, or the sides of the ladder, if you will, are made up of phosphates um, and a 5-carbon sugar called deoxyribose. So phosphates and deoxyribose form the backbone. So what about the steps or the rungs of the ladder here, if you will? So these are made up of molecules called nitrogenous bases, all right? And there's 4 of them found here: A, G, C, T. A for adenine, G for guanine, C for cytosine, T for thymine, okay? We talked about those before too. And something else, something that is called the Chargaff rule. He was another scientist, Chargaff, I believe it was Russian or Eastern European. Chargaff rule. And that states that A always binds with T and G always binds with C. So remember, apples on a tree, A with T, cars in a garage, G with C. And that's exactly what you see. You see that G goes with C and A goes with T. So that is how the DNA is structured, okay? Um, and it's an unbelievably long molecule. And so the estimates are, if you were to untangle all the DNA in all the cells of a single individual, you could go from here to the sun and back 1,200 times, more than 1,200 times. Unimaginable. That much DNA, genetic material, crammed into a single human being, okay? It's all there. So that gives you the basic idea as to what the DNA molecule basically looks like. So what are genes then? Genes are those portions of your DNA that code for a protein, that code for a certain type of protein to be made in your body, like your eye color protein, skin color, melanin, or keratin, which is the protein that forms your skin and hair. So those are basically synthesized based on chunks of information from parts of these DNA. All right. All right, so what is the function of nuclear pores within the nuclear envelope to allow for messenger RNA to move in and out of the nucleus? What is the function of the nucleolus? It contains ribosomal RNA and protein. The structural relationship of DNA and chromatin. Chromatin is this untangled, all tangled, not untangled, this entangled, all tangled up bunch of DNA. Kind of looks like Christmas lights all tangled up, lying on the floor. That's chromatin, right? And the relationship of DNA and genes, the protein coding portions of DNA are called genes. They actually make proteins for your body. Okay, so here we are looking at what is called the central dogma of life. So let's write it down here. This is the central dogma of biology, all right? So what do we mean by the central dogma of biology? It simply means DNA is translated, or rather transcribed into messenger RNA, which is translated into protein. 3-step process, right? From DNA to messenger RNA to protein. And the conversion of DNA, or transcribing of DNA into messenger RNA, is called transcription. which I'm writing down here in my notes, and then the translating of messenger RNA into protein is something called translation. So this three-step, or rather two-step process, transcription and translation, as you see here, is called the central dogma of life, okay? Okay, so let's go ahead and take a look at what we actually mean by all of this, all right? How does this whole thing work? All right. So I'm going to basically summarize all of this for you so that it becomes quite easy, hopefully, for you to understand and all the other complex steps involved here. So here goes, let's start. So now I am going to unshare my screen, stop share, so you can get a better view here, all right? Okay, so let's see. So what I'm going to do is, and this might be a problem because the pen stops writing if I'm holding it up. So let me see if I can use well, let's see. Let's try. All right. Yeah, just as I thought. Okay, so let me get a marker here. Okay, so here, better. So I am going to draw a DNA molecule inside of a cell, right? Hypothetical situation. And let's fill it up with letters, right? The nitrogen bases. Let's make some up here. Let's say A, G, T, C, G, A, T, C, G, okay? Um, and so of course, once we know these letters, let's make a strand farther over here. We can fill up the other side as well, right? So, all right, this is better. Let me get out of the picture here. Okay, so now I am going to write down the corresponding letters next to them, right? So the A goes with what? T. I'm going to write a T here. G goes with C, T with A, C with G, G with C here, A with T, T with A, C with G, and G with C, right? So these are the two strands of a DNA strand, okay? So when the first step of central dogma of life, which is called transcription, happens, what happens at that point is the two strands separate out. The two strands separate, they open up, and they open up by means of an enzyme called DNA helicase, all right? Helicase. It's like the unzipping enzyme. It unzips the DNA molecule. Since it unzips the helix, double helix, it's called DNA helicase. So it makes sense, right? Okay, so now, uh, and this is all happening within the nucleus, of course, because the DNA is found inside of the nucleus. We know that, right? Okay, so once it has unzipped, let me redraw this strand here, strand of DNA, A, G, T, C, G, A, T, C, G, right? So this is the unzipped original part of the half of the original DNA molecule. Okay, so what happens next in this step is that another molecule lines up right next to it, okay? This was our DNA molecule, half of the original DNA molecule. But the other molecule that is basically lining up next to it is a messenger RNA molecule, okay? In other words, this is transcription in action. This step is called transcription. And where does transcription takes place? It takes place inside the nucleus, in the nucleus. So I'm writing all of that down here, okay? All right, so let's fill in the letters then. So what goes with A? You would say T, correct? Remember though that RNA does not have a T. RNA instead has a U for uracil, something to remember. And since this is messenger RNA forming a template or a copy of the DNA, so you will write a U here, right? Okay, then what comes next to G? A C. Then what comes next to T? An A. What comes next to C? G again. What comes next to G? A C. What comes next to an A? Not a T, but a U. Remember that there's no T in our mRNA. What comes next to T is an A. What comes next to a C is a G. And what comes next to a G is a C. So here is a complete transcript of the DNA molecule copied by the messenger RNA. And notice how there are U's instead of T's in the messenger RNA, okay? This is called transcription, and transcription takes place where? Inside of the nucleus of the cell. All right, so far so good. So now what we have done is you have transcribed the DNA, the genetic information of your DNA molecule in terms of messenger RNA. So why is it called a messenger RNA? Because it takes the message, right? So it's going to take the message, which is this encoded genetic information, the sequence, and it's going to swim out of the nucleus, right? It's going to swim out of the nucleus, and now it gets into the cytoplasm of the cell, and it finds the nearest ribosome, which I'm going to draw here. Draw this sausage-like shape here, called, that's a ribosome, right? Ribosome. And since the messenger RNA traveled all the way out from the nucleus, it might be mighty tired uh, uh, after all this journey. So it's gonna just kind of lie down here on its back. All right, and let's write down the same letters that it got from the, from where? From the DNA, right? So what was that? It was U, C, A, right? G, C, U, right? And then A and G and C, right? So it's the same information that it got from the DNA in the nucleus, all right? So something magical happens after this. Another molecule starts approaching here and joining to our messenger RNA here, okay? All right, this messenger RNA molecule. So let's label, just that everything is clear, let's call it messenger RNA. Again, this is messenger RNA. So this molecule that comes into place actually looks like it's almost like a shoe plate shape, right? Or a flying saucer or some, whatever you want to call that, right? So it has this peculiar shape like this, okay? This shape right here, okay? All right. So these three weirdly shaped molecules are coming and they're trying to attach to the messenger RNA here. And what are these weird shaped molecules? These are tRNAs for transfer RNA, okay? tRNA. These are tRNAs. Now, these tRNAs, these ones are trying to attach here to the messenger RNA. But how do they attach? Let me quickly check if hopefully I'm, okay, it's recording, right. So how do they attach in place? So these need to read this genetic code here in triplets, in 3 letters, right? So for example, this one has to attach to UCA, these 3, all right? But in order for this tRNA to attach to this chunk of messenger RNA, so this triplet first, UCA, it's called a codon. Okay, so these are codons, UCA, GCU, AGC. These are all codons. So let's call them this, right? Let's call them codons here. CODONS. These are all codons, as you can see, okay? So in order for this tRNA to attach to UCA, what 3 letters do you need here at the base to go with these, to click in place, right? So again, what goes with U? An A. And what goes with a C? A G. And what goes with an A U, because there's no T there again on tRNA either. All right, right? So AGU, as you can see, AGU is what's going to click into place. So this AGU is called, since these were called codons, so this corresponding one is rightly called anticodon. Okay, so let me label these 2 here. So these are called anticodons. There you go. Anticodons, all right? So let's crack the next one. This one has to go with what? With GCU. So what 3 letters do you write here? What goes with G? C. What goes with C? G. What goes with U? A. So CGA CGA is the anticodon that goes with this codon. And then finally you have what? AGC. So what goes with AGC? Let's see. What goes with A is U. What goes with G is C. And what goes with C is G. So UCG. UCG. That's your final anticodon. All right. So far, so good. So these tRNA are clicking into place onto the messenger RNA. All right. But how does that make protein? Well, on the other end of the transfer RNA, on this knobbly head at the top that you see, there's a specific type of amino acid attached. Let's say this one has an amino acid called isoleucine attached to it, right? Just as an example. The other one might have an amino acid called methionine attached to it. So let's call it methionine. The last one, let's say, might have an amino acid called uh, asparagine, right? Well, let's call it yeah, there you go. Asparagine attached to it. So there. So what just happened? Based on this information, which was which were the codons that the messenger RNA had actually copied originally from the DNA in your nucleus, you are building these amino acids which make up proteins based on this information. Eventually, these tRNA are gonna get away from under here, and these amino acids are gonna click together, forming a long amino acid protein chain. And so this protein formation process here on the ribosome in the cytoplasm of the cell is called translation. Translation. All right. So that clarifies what that was all about, translation. Okay, there. So let's quickly recap what happened, right? First step, we are looking at transcription. How did that happen? Here was a DNA strand inside of the nucleus. Those were the letters. The enzyme called DNA helicase came in. It unzipped the two parts of the molecule. Here's one portion of the DNA. So then another molecule started gathering up in front of it. And this was a molecule of messenger RNA, and it formed a perfect complement. No T here, only U's, right? This is called transcription. There's also an enzyme involved here. When this chain was forming, there are other enzymes involved too, like this enzyme was called RNA polymerase. Makes sense because it helped form the RNA change, right? Chain. So RNA polymerase, okay? Anyway, so after you form this messenger RNA chain, it swam out of the nucleus, went to your ribosome in the cytoplasm, and here it lined up. Here is your original genetic information, right? And then so that, and we are studying these letters in threes, in triplets called codons, right? And these attracted these transfer RNA molecules, which clicked into place using their anticodons, these sequences, and each of these transfer RNA was carrying an amino acid like isoleucine or methionine or asparagine on their backs. And so these clicked into place, and now you have a long protein chain, which might give you your blue, wonderful dad's blue eyes or hazel eyes from mom or something, based on the genetic information in the DNA coded there. So transcription in the nucleus and translation on the ribosome in the cytoplasm right here, okay? That is it. So those two steps, transcription and translation, are together called the central dogma of life, okay? As we mentioned here, central dogma of biology, as you see, okay? Now, something else to remember? Well, this was an example, right? But usually, always, this amino acid chain or protein chain starts with a methionine. Methionine, okay? So that's something to remember. Methionine should come first. Just something to keep in mind. Also, before this messenger RNA can exit the nucleus to go to the ribosome, there's at least three processes, something called mRNA processing that must take place before the messenger RNA is allowed to make its exit and go to the ribosome. All right, so what are those three steps? Number one is called what is it called? Guanine capping or G capping, okay? Let's write down that first, guanine capping. The second step that must happen is something called the addition of the poly A tail. And then finally, what is called exon splicing. Okay, 3 steps which make up what is called mRNA processing. So what is each? All right, let's take a look at the first one. Guanine capping. What does that mean? Before this messenger RNA molecule can get out of the nucleus, a G cap. So let me draw a cap here. All right. Here's a cap, and it has a G logo on it, right? So a cap of G or guanine has to be added to one end of the molecule. All right. And this is called guanine capping, the G cap, you can see. And then a poly A tail, which means I'm going to draw it here too. To the tail of this messenger RNA, you're going to attach a long tail of A's, and those A's are for adenine. Okay, so this is your here. This is your poly A tail. Poly A tail. So you put a guanine cap on the top, and then you added a poly A tail to the other end of messenger RNA. Okay. So the final step that must happen before it can leave the nucleus is something called exon splicing. So what is an exon? First step, right? Exon and then something called intron. So exon are those chunks of the messenger RNA which are snipped off. They're cut out and they do not make it out of the nucleus. Okay, so those are called actually those are called introns. All right, so introns are cut out. Introns are cut out. They just stay into the nucleus. They don't make it out. So introns, right? And the parts that remain are called exons. And because they exit the nucleus, therefore they are called exons. And so then those parts are joined together, which is called splicing, exon splicing. And then this molecule is ready to move out. Okay, so again, guanine capping, poly A tail addition, and exon splicing. Again, exons are those portions of the messenger RNA which would be allowed to exit the nucleus. The introns are cut out, no introns, right? So it's introns, they're gotten rid of. So no introns, okay? No introns. Exons make it to the outside. So that's pretty much it. Transcription, translation, all right? And the pre-mRNA or the mRNA processing that you see here, 3 steps. Okay, so hopefully that made some sort of sense to y'all, okay? And let's go ahead to, where were we? There, okay. Right, so I'm gonna go back all the way here. All right, so there. All of these things, transcription, translation, right? The 3 types of RNA, we talked about those as well. How does transcription take place? You can see it here. mRNA processing, splicing, exons, introns, guanine capping, poly A tail, translation, right? Codons, anticodons, you can see all of those in the picture here. Okay, that's all you needed to know. So DNA is the control center of the cell, also called the brain of the cell, as you can see here. All right, what are the 3 major structures required for transcription? What do you need? You need a DNA template, you need messenger RNA, and you need RNA polymerase. Explain where and how transcription occurs. It occurs in the nucleus. And how does it occur? It copies the DNA. We just looked at it. What are a codon? The 3-letter triplets on messenger RNA. What is an anticodon? The corresponding 3-letter triplets on the transfer RNA or tRNA. How is messenger RNA attached to ribosomes and translated into the language of protein? That is called translation. And we went over how the whole process of how codons and anticodons click together. And on the back of each transfer RNA molecule, you have a specific amino acid. The genetic code of DNA is the specific instructions to make what biological molecule? Proteins. They make proteins, right? Eye color, hair color, all genetic traits are proteins. Okay, next up, we are looking at cell division, which is called mitosis, all right, which is one type of cell division. There's also something called meiosis. So mitosis is how your cells divide in your somatic cells, in your non-sex forming like non-sperm, non-egg cells, okay? Meiosis is another type of specialized cell division that only takes place in sperm and eggs, in your testes and ovaries when making sperm and eggs. So that's something to remember, okay? So what are we looking at here? We are looking at the cell cycle, okay? And the cell cycle is, it looks like this. So a major portion of your cell cycle is basically spent in something called interphase. So 23 hours, look at that, out of 24 hours total, 23 hours are spent here during interphase. Interphase is also called the resting phase, but the cell is not really resting. So it's kind of like a misnomer. The cell is quite active here, okay? And it's doing things like replicating its DNA, doubling the DNA, making repairs and all those kinds of things. However, the interphase is divided into these three subphases, the G1 phase, the S phase, and the G2 phase. So what happens during the G1 phase? Growth and preparation for DNA replication, those kinds of things. S phase, so you can think of this S as for synthesis. Synthesis phase or S phase. This is when the DNA doubles itself. The DNA of the cell doubles itself, right? And then you have the G2 phase, and what happens during the G2 phase is, again, growth and preparation of division of cell. Interphase is not part of mitosis. This is pre-mitosis, before mitosis. After the cell has gone through all these steps, then it enters into the mitosis stage. Mitosis is very short. It's about only an hour out of 24 hours. 23 hours is interphase, only 1 hour mitosis. Mitosis has these three stages: prophase, metaphase, and 4 phases. Prophase, metaphase, anaphase, telophase, and then something called cytokinesis, when the cell actually splits into two identical daughter cells. Okay, that's what the cell cycle or mitosis is all about. Okay, and so that's basically what we are looking at here. Talked about, so interphase, this is what the cell looks like under the microscope during the resting phase. Interphase, which itself is made up of G1, S phase, and G2 phase, and then the 4 phases of mitosis. All right, and how you might remember this is by memorizing the acronym IPMAT, IPMAT, right? If you remember IPMAT, then you will remember that I is for interphase, and remember, interphase is not mitosis. It's only preparation for mitosis. Then P is for prophase, right? M is for metaphase, and A is for anaphase, and then T is for telophase, okay? So let's quickly go ahead and see what happens in each of these stages, all right? Okay, so first up, we have prophase. Pro means before. So this is the first stage that happens. It's also the longest phase of mitosis. So what happens in prophase is the nuclear envelope, the nuclear membrane around the nucleus dissolves or disappears. The chromatin condenses into chromosomes. Chromosomes are these thread-like structures that contain your DNA or genes. And these two centrosomes or centromeres start to appear at the 2 poles of the cell. That's what happens during prophase. Then comes metaphase. So think of metaphase as the letter M for metaphase. The same letter M could stand for middle. So your chromosomes actually line up in the middle here. They're lining up in the equator in the middle. And you see how the centromeres are sending out these thread-like fibers called spindle fibers. And the spindle fibers attach to the central point of these chromosomes here. So this is metaphase, all right? Then comes anaphase. Anaphase starts with an A, letter A, and you can think of this A for away or apart. So these spindle fibers they've split the chromosomes into 2 daughter chromosomes or sister chromatids, and each is being pulled to the opposite pole. They're being pulled apart. And then finally telophase, T for telophase, T for 2. Now you see how it's splitting in the middle, it's pinching in the middle, and eventually it's going to separate out into 2 genetically identical clone daughter cells. And then cytokinesis. Cytokinesis is when the 2 daughter cells split. Each daughter cell receives exactly the same amount and number and type of genetic material as the parent cell. This is cloning again, right? For example, in humans, we have 46 chromosomes in each body cell. So the 2 daughter cells that form out of each cell also has 46 chromosomes each, okay? And how does that happen, even though it's splitting into two? Because remember, during interphase, the S phase, the DNA doubled itself. So that's why when it splits into two, it gets back to the 46 chromosome number. So how is chromatin distinguished from a chromosome? Chromatin is like tangled up Christmas lights type of appearance. Chromosome are well demarcated bunches of DNA. Describe the process of DNA replication that occurs during the S phase of interphase. DNA duplicates itself. What are the events that occur during the mitotic phase, mitosis? So IPMAT, remember the IPMAT, right? Or PMAT for mitosis alone. So then aging, what happens with aging? Your cells start to die, all right? That's what's happening with aging, as you can see here, either because of exposure to ultraviolet radiation or alcohol or cigarette smoke or simply aging, like you have been in this world for that amount of time. So your cells start to die by a process called apoptosis. This is programmed cell death. This is natural. With time, some cells, they commit suicide, like using lysosomes, right? So that's apoptosis. And initially, apoptosis is there for repairing your old worn out cells, but as we age, apoptosis takes place throughout the body and even healthy cells start to die, okay? So this is what happens to your DNA. It starts to break down too with time. Your DNA polymerase enzyme is destroyed. The DNA gets digested away, right? This also makes you prone to developing cancer. Older people develop more cancers because their DNA is being going all haywire and being destroyed too, as you can see here, all right? So apoptosis is programmed cell death. Interestingly, when you were in the womb, still developing, our fingers and toes were actually webbed. There was webbing like frogs or something, right, in between our fingers. And by about 7th month of pregnancy, thanks to apoptosis, these webbing cells, webbed cells actually destroyed themselves, so our fingers opened up. And some evolutionary scientists use this as proof of our descent are from sea creatures, from marine. So we literally crawled out of the ocean, and we share that ancestry with our marine relatives, right? So so webbed fingers slash toes and apoptosis. And in some preemies, children that are born prematurely, you can see these webbing. The webbing is still there, right? So which would be an advantage if there were swimmers or something. And we probably have this webbing because we're in the amniotic fluid. It's kind of like a waterful bag in which we are suspended. It helps us move around and those kinds of things. So it's quite fascinating. All right, so then tumors. What are tumors? Tumors literally means a swelling. So when your cells keep on dividing, that's called a tumor, right? A tumor could be benign or it could be malignant. How do you distinguish the two? Benign tumors are limited to an area. They do not disseminate. They do not spread to the rest of the body. Malignant tumors slash cancers actually can metastasize, which means they can break off from their place of origin and they can travel with your bloodstream, with your lymph flow to far and wide reaches of your body and put little seedlings there and develop new cancers. So that's what tumors are. So what are the specific changes that occur to DNA during apoptosis? It basically self-digests, it breaks down, turns into little blobs. DNA polymerase enzyme is destroyed. So your chances of developing tumors, both benign and malignant, and of course your chances of dying go up substantially. All right, so here we are done with chapter 4. This one was rather long. So again, remember, turn in your lab work. I'm going to send out that on a daily basis, even though I'm going to grade it on a weekly basis. So I mentioned that in my email. So keep up with the study guides. Shortly, I will also be sending out your case studies guidelines, how to work on those. So keep your eyes peeled for that one as well. Keep your Connect lecture quizzes in mind. Tuesday is your first upcoming lecture exam number 1. I'm hard at work preparing your study guide for your lab exam number 1, which is next Monday after this one. So I'll be sharing that too. Okay then, so enjoy the rest of your weekend and have a fantastic week ahead of you. And I'll see you all virtually tomorrow with a discussion of chapter 5. All right, bye-bye.